So my goal for today is to talk about synchronizing slow oscillations, the big slow waves of deep sleep, with the goal of extending deep sleep and really enhancing the depth and duration of stage N3, the deep stage of sleep. And we are the Brain Electrophysiology Laboratory, and we've created a new company called Neuroscience, a corporation to build medical devices specifically for sleep assessment and therapy. And, and so we have a strong interest in aging and dementia as well. So I think, as you mentioned, there's quite a bit of overlap between our efforts and interests and the work in your lab, and I think that should become clear as we go through this. So the Brain Electrophysiology Lab was first established in the University of Oregon many years ago. We became EGI, focusing on high density or dense array electroencephalography. And we retained that focus. We sold EGI to Philips a number of years ago, and then established Bell really for advanced software development. But because Philips kind of failed to carry forward with EGI, we've now developed a complete hardware system. And so you'll see that in the course of our science work that I'm going to emphasize here. And I have kind of an ambitious set of goals that I've put into the slide for looking at how to synchronize the slow oscillations of deep sleep. And I won't go into all the detail now, just so we can get through the talk in a reasonable time. But the main points are, we want to understand the slow oscillations neurophysiologically. And there, the high density helps us to understand the sources of the slow oscillation of deep sleep, which we do think are critical for normal memory function. So that when the slow oscillations decline with aging, then there is a corresponding decline of memory. And of course, eventually it becomes catastrophic in dementia. But as we're going to show, the decline of deep sleep and the loss of memory are normal neurodevelopmental processes of aging. They may not be attractive or beneficial, but they are actually developmentally predictable. But our goal is to reverse the decline of slow oscillations and to improve the synchrony of slow oscillations with applied external electrical current. So I'll show that effort and some of our recent success in doing that. And let me just emphasize that please break in at any time. I really haven't practiced this talk, so it's going to be informal and easily interrupted if I'm not clear about something or you want to go more in depth. And I've tried to structure it for a half an hour so that we would have time for discussions and interruptions. Here's our current high-density EEG system, the Bell System 1. It's a 280 channel advanced geodesic head web, and a number of the advances we've made allow us to do recordings all night in sleep, including at home. And part of the goal of making this portable and easy to use was to make the high density amplifier battery operated and wireless, so it could link to the home internet and provide two-way communication with our cloud server. The question really for high density EEG is source localization. How do we understand brain activity, not just EEG signals at the head surface, but the sources, the generators, which we think are almost certainly cortical sources. So we want to map the electrical fields of the cortex, and the basic physics are that those electrical fields are perpendicular to the cortical surface. And because the cortex is wrinkled, we have to understand those wrinkles and actually quantify them exactly so that the dipoles that we're localizing, the dipolar fields, must be perpendicular to the cortical surface. And you can see, given the individual wrinkles that we have, the best localization will be with an individual head model where we can characterize those wrinkles. And the perpendicular electrical fields of the cortex with a high density electrical source localization. So we've worked on this problem for many years, and just briefly, our main approach has been to improve the anatomy that we're localizing to. And this includes the cortical surface, as I've tried to emphasize, but also the conductivity of head tissues, where the skull is a particular problem. So we want to know the dipoles, and you can see the region of dipole placement. All of the dipoles, for example, we may have 2400, 4800, or now 9600 dipoles that we're attempting to compute from the inverse of the 280 channel data. But their orientations need to be considered, and the intervening tissues that impede the electrical current getting to the surface electrodes need to be understood. And so the cortical surface has to be extracted, and we have to characterize each of the tissues, including the skull. So if we don't have a CT, and usually we do not because it involves ionizing radiation, we fit an atlas CT that's close to that person's head shape, and then we warp it to their MRI, specifically their T1 MRI, so that our computational model of the electrical inverse includes all of the conductivity of the tissues. So we integrate those within our software package called Sorcerer. So Sorcerer is really the best anatomically specific EEG source localization package because we've accounted for all of these factors. So the other fact was to give good visualization of the EEG on the cortical surface, but also to have a user interface so that each user could be trained on these basic physics of how cortical sources generate electrical field, which is seen in the EEG. And finally, we've wanted to integrate this within laboratory operations that could include high performance computing, database archiving of the EEG, and individual analytic routines where each lab could build its own unique processing workflows. And we call this the forward-looking operations workflow or flow. So let me illustrate high-density EEG and the perspective it can give on underlying features. And I'm going to focus on the large, slow oscillations of sleep, often called slow waves. And either oscillation or slow waves kind of misrepresent these phenomena, these large, slow oscillations, because they're not oscillations in the sense of sine waves. They can look like that to kind of a low density EEG, not studied very carefully. But actually, it turns out that each large slow oscillation is a transient. It's not really a sinusoidal feature. And you can see it in the high density EEG over here. The brown line in the EEG shows a single punctate event, a slow oscillation. Actually, you could also call this a K complex because they share a lot of features. The main feature is a big negativity that's typically toward the front of the head, not always. And it's followed by a smaller positivity, a second waveform that looks to be kind of a reversal of the first big negativity. Now, one of the things, if you don't understand source localization, you might describe this as a frontal negativity. and you might ignore the positivity in the back of the head. And in fact, a number of leading labs have done just that. And so they saw the big negative field moving over the head as the source actually changes, and described it as a traveling wave that was in the frontal lobe. And this actually turns out not to be the case for reasons you can see if you look at the head field distribution on the right. Exactly coincident with the frontal negativity is a posterior positivity. That suggests the source is dipolar. And in fact, if you look at the inversion between the negativity and the positivity, you see this is in the midline, middle regions, such as in the temporal lobe. And in fact, that is the source. That is the generator of this field, not the negativity or the positivity. Now, that comes out naturally when you conduct an electrical source analysis, an inversion to have a putative cortical source for creating the surface fields. So that's what we do in Sorcerer. We do this source inversion. And on the left, you can see the big negativity. So this is 40 channels taken from the front of the head. So everything looks negative. But if you plot the head field distribution, as in B here, you see that there's a frontal negativity, but a posterior positivity, exactly coincident in time, suggesting that the source is somewhere in the middle of the head, and particularly around the temporal lobe, the anterior to medial temporal lobe. And this is what our source localization with the Bayesian multiple sparse priors, the best algorithm that we found for source inversion shows. So in C and E, you can see that the source that explains this head field distribution is actually in the right medial temporal lobe. And this is a pretty tight focal source in the parahippocampal gyrus. And it's not untypical of the people that we look at. So the main point is that slow oscillations do look very large and impressive, but they're not diffuse across the head. We think there are up waves and down waves, up states and down states that are local in the neocortex during the slow oscillations. And there's plenty of intracranial recording that suggests that. But those are fairly small field patterns that are difficult to localize in the EEG. The major EEG events we see are in limbic regions. Here's another person. Again, if you look at A, you're seeing the voltage field patterns in the frontal areas that are negative going. We have the typical frontal negative, posterior positive pattern, suggesting an anterior temporal source. And then here, if we do the source localization, there is activity in the anterior temporal, but also the caudal orbital frontal, here, area 25. That's, again, a typical generator. The other feature that's interesting here is that when the wave inverts, when the frontal negativity kind of repolarizes or whatever it does, there's a field inversion and a frontal positivity follows. And if we localize that, it shifts from a source in the caudal orbital frontal to a sink. So there is a reversal in the following positive wave. But by and large, in the study that we did, this is Morgan et al. In Sleep Research 2021, very typical. The generators of slow oscillations are in limbic areas. And we can make a flat map to characterize this. So for example, if we look at the count, the number of slow oscillations localized to each brain region, and the top image shows the Brodmann areas of the cortex, the bottom area shows the density or the number of counts of slow oscillations over this sample of 20 individuals that we localized. The density is strongly in limbic regions for the slow oscillations of deep sleep. Now, part of our interest is to understand why. What does it mean that there are limbic generators? And how do we understand this neurophysiologically, but also functionally, to the functions of slow oscillations and deep sleep in memory consolidation? Because the evidence is quite good that normal consolidation of memory requires the non-REM sleep, and particularly the stage 2 and stage 3 features of spindles and slow oscillations in order to consolidate explicit memory. We're very interested in the neurophysiology that suggests that slow oscillations are synchronized and coordinated by the claustrum. And this might help to explain why we see the strong ventral limbic control the amygdala and orbital frontal cortex as being the primary control systems for the slow oscillation of deep sleep, because the amygdala claustral complex seems to be involved in the widespread synchronization. And with widespread projections from the claustrum to limbic cortex as well as neocortex, this might be a way of understanding the neurophysiology. And then the function seems to be important, excuse me, not only to normal memory consolidation, but also the decline of memory that occurs with the loss of deep sleep in aging, and particularly in Alzheimer's. Excuse me. So I'm pointing now to the classic neuropathology of Brock and Brock, showing that as Alzheimer's progresses, the neurofibrillary tangles and plaques accumulate in limbic regions. And this really clearly appears to relate to the memory impairment that happens in Alzheimer's disease. So the link between slow wave decline in aging and dementia seems to point to these limbic regulations of memory consolidation in the normal brain, so that when sleep, slow wave sleep particularly, declines in aging, there is a loss of the memory consolidation and therefore memory impairment. This is a recent study I'm pointing to from the Framingham Heart Study that looked at normal senior citizens, normal old people without MCI with over time. And what they found, which again is a normal progression, the ones that lost more of it were at risk for dementia. And in fact, in this remarkable ratio, that a single percentage decline in a year predicted 27% increase in the risk of dementia over the subsequent years at that point. So we're very interested in understanding the nature of these mechanisms and how to appreciate them. Here I've plotted the amplitude of the slow wave sleep, and you can see the normal developmental course involves a decline in adolescence and a continuing decline in aging. This is from our own machine learning studies that we applied to the Cleveland Family Studies database available at NCRR. And you see the decline in adolescence is quite remarkable. It's a normal neurodevelopmental process to lose deep sleep. But then about age 30, there's a further decline for many people, but not all. Some people maintain their deep sleep in aging. Many people do not. And we think the evidence points to the ones that lose it are the ones who are on the road for cognitive decline in aging. The question is, how could we improve this? This became really the goal for Neurosom and creating a medical device company that could use a simple headband to synchronize the slow oscillations of sleep. What we did was to reverse our computational modeling. So now we're not trying to localize the sources of the slow oscillations. We're trying to inject current, to apply current into the head in an optimal pattern for targeting these deep limbic regions that seem to be the sources. And it turns out that when we do this computational modeling, what we find is that we don't need the high density array to have a pretty reasonable targeting of the deep sources. And in fact, if we choose the optimal electrodes, we can achieve a fairly good targeting of the limbic generators, anterior temporal and caudal orbital frontal particularly, with just a few electrodes. And that's what this simulation shows. On the left is a simulation of the original TES synchronization of slow wave sleep by Lisa Marshall in her group, using fairly high current levels of like a milliamp. And that's enough that it's a little bit noxious. What we showed on the right is we can have a fairly good approximation with half of that current. So the question was, if we cut the current and only use half of a milliamp or 500 microamps, could we still synchronize the slow oscillations of deep sleep? And that's this study, Hathaway et al. We targeted the sleep stages, and here it was before we had our machine learning that was working adequately. So what we did was have a research assistant looking for N2, and as you know, you can see the spindles and K complexes of N2 are pretty easy to detect by visual inspection. After 4 minutes of N2, we judged that the person would be typically ready to go into N3. We turned on the stimulation, again manually. And what we found was an increase of N3 that was small but highly significant. In this study, it was 17% increase in N3 on average. And interestingly enough, it not only increased during the stimulation, but it was stronger than those in the sham group throughout the night, the other cycles of N3. So the indication was that it was fairly reasonably easy to synchronize the slow oscillations. And our goal is to understand the pathophysiology of the N3 loss and what it means for aging. Now here I'm showing an example of the clearance of toxins such as amyloid beta and tau that is achieved by the brain's glymphatic system. And there's fairly interesting evidence that the brain's glymphatic system is strongly active during deep sleep, during N3 sleep. Some of the evidence actually came out of MGH, Laura Lewis's lab, where they focused fMRI actually measures to assess the CSF clearance by looking at the base of the brain and to estimate the clearance of CSF. And what they found, shown in figure E here, or patch E, is that during slow wave sleep, there are big fluctuations, and you might think of it as pumping CSF out of the brain, such that, for example, in figure G, there is a much higher increase of the CSF excretion from the brain or clearance from the brain in sleep than in waking. And this appears to be specific to the deep sleep. Our goal then was, how can we enhance the synchronization of slow oscillations, the electric sleep therapy, as we call it, at home? Because we know that sleep in the lab is somewhat unusual. It's not like sleep at home. So our goal would be to have a sleep therapy at home. Here's one of our first prototypes. It's not particularly elegant, as we wanted to bring the whole technology of a battery operated method for synchronizing TES. That's not so hard to do. But we also wanted to integrate it with machine learning. And it turned out that's not so hard to do these days. We have worked with convolutional neural nets for a decade, and the hardware keeps getting cheaper. So NVIDIA has some remarkable chips that allow us to implement TensorFlow and other machine learning for our convolutional neural nets to detect the sleep staging in real time. And I think you guys are familiar with that from your own work. So we set up a nanocomputer at the bedside, and we interfaced it with our simple Bluetooth headband. And this became really our medical device delivery to build from our high density EEG to make a simple headband to do EEG classification, and then TES therapy for synchronizing deep sleep at home in an automated closed loop package. And that's really the framework for Neurosom, the company that we've now established to bring this to medical devices with FDA clearance for specific indications. And our first indication will be healthy adult sleep, because that's really who needs it. It's not after you have MCI or cognitive decline or dementia. It's kind of too late then. It's when you're still healthy, but getting old. That's when the deep sleep therapy is necessary. So that's really the framework for Neurosom. And I will mention that if you're interested in investing in Neurosom, I have a brief commercial here, that we are available for investments right now on the Red Crow platform. Here's some of the results of our first home sleep study. So our first goal is to say, okay, we think an understanding of the slow oscillations of deep sleep and how to target them most effectively. Let's prove it. And let's start with healthy adults. So here we have a range of healthy adults. A lot of these at this stage of our kind of preliminary analysis are younger adults, so they're pretty easy to work with. But they're also equivalent to the Morgan et al. And Hathaway et al. Samples or earlier published work in they're fairly young. Not all university students and recent graduates, but a lot of them. A few older people that do have good N3 synchronization, but we need to improve the aging sample. What we find is that at home, people sleep much better, and the N3 enhancement is much better. So rather than a 17% with this first interim analysis of the home sleep, we get a 44% increase in N3 enhancement. So it's not so hard. It works fairly well with healthy adults. We have funding now from NIA, National Institutes of Aging, to bring this to adults with mild cognitive impairment. And we're working with Ruth Benka at Wake Forest University and with Miranda Lim at Oregon Health Sciences University. In a phase 1 trial, we were able to train the seniors with MCI that we recruited from the Wake Forest, Winston-Salem, and Portland, Eugene community, with help from family members in many cases, to use the sleep wisp at home. So we showed we could get good data in a home environment with this technique. So we do want to bring it to aging as well as to bring it to healthy adults that we think is a major target. And the goal is to improve glymphatic clearance, to improve the washing out of the toxins. Amyloid beta and tau are kind of the major ones, and their downstream effects on neurodegeneration, which can be indicated by glial cell measures such as GFAP, and the neuroinflammation that's part of the pathophysiology. And here we've teamed up with John Nailway and his colleagues at Sonus Biotech, also located in the research park at the University of Oregon, right next door to us, to measure markers of the glymphatic clearance. And we're particularly interested in doing frequent measures, even daily measures, with nose sponges and nose swabs, because the nasal secretions seem to be a very good route for routine daily assessment. So that's really the overview of our research program. We have basic science interest in understanding the mechanisms of slow oscillation, how they figure in memory consolidation, and how they can be maintained at healthy young adult levels as people are aging and moving on to the later years of their life. So I'll stop there and open up for questions at this point.